My name is Antoinette Evangelista Fernandez. Today I am going to present my presentation about the seed morphology and seedling structure. So seed is a small embryonic plant enclosed in a covering called the seed coat, usually with some stored food. It is the product of the ripened ovule of gymnosperm and angiosperm plants, which occurs after fertilization and some growth within the mother plant. So, the internal and external structure of corn and mung bean seeds. So, this is the external structure of corn, which is the endosperm, pericarp, germ, and tip cap. Then the internal structure are the endosperm, cotyledon, seed coat, radicle, hypocotyl, epicotyl, and the embryo. Next is the mung bean. The external part of mung bean are the micropyle, hilum, seed coat last, and the internal structure of mung bean are the hypocotyl, epicotyl, radicle, seed coat, cotyledon, and embryo. Next is the functions of the part of the seed. First is the internal part of corn, which is the epicotyl that directly responsible for growing in order to punch through the surface of the ground. Then hypocotyl pushes the cotyledons above the ground to develop. Then radicle that allow the seed to suck up water and send up its leaves so that it can start photosynthesizing. The seed coat that protect the embryo and to transmit information regarding the external environment. Then cotyledon that may aid the embryo in metabolizing nutrition stored elsewhere in the seed. Then embryo that well known that embryo secretes signal to the endosperm to induce the degradation of the seed reserve and to promote endosperm wicking during germination. Then endosperm supplying nutrients for embryonic growth and providing some amount of protection for the embryo. Next is the external part of corn, which is the pericarp that protects the enclosed endosperm and embryo from attack by fungi and bacteria. Next is germ, only living part of the corn kernel. It contains the essential genetic information, enzymes, vitamins, and minerals for kernel to grow in a corn plant. Then tip cap, the attachment point of the kernel to the corn cob, where the nutrients and water flow, and also it contains fiber. Then endosperm, supplying nutrients for embryonic growth and providing some amount of protection for the embryo. Next is the internal part of mung bean, the epicotyl, directly responsible for growing in order to punch through the surface of the ground. Hypocotyl that pushes the cotyledons above the ground to develop. Then radicle allow the seed to suck up water and send up its leaves so that it can start photosynthesizing. Then seed coat protect the embryo and to transmit information regarding the external environment. Cotyledon that may aid the embryo in metabolizing nutrition stored elsewhere in the seed. Then last is the embryo that well known that embryo secretes signals to the endosperm to induce the degradation of the leaves reserve and to promote endosperm wicking during germination. Then last is the external part of mung bean, the hilum, a scar on the surface of the core formed when the ovule detaches from the ovary wall. The seed coat that surrounds the embryo and protects it from damage and against attack of microorganisms such as the bacteria and fungi. Last is the micropyle, a hole through which embryo absorbs water before it germinates. Then the stages of germination. First are the germination of mung bean. This is the germination after 1 day. Next is the after 2 days. Then after 3 days of germination. And after 4 days. And last is after 5 days of germination. So that are the 5 stages of mung bean germination. Next is the germination of corn after 1 day, after 2 days, and after 3 days, after 4 days, last is after 5 days. So that's are the 5 stages of corn germination. So next is the parts of germinating seed. So the parts of the corn. Next is the parts of the mung bean, which is the leaf, cotyledon, stem, and root system. Then last is the documentations. First step is the pre-soak 5 to 10 seed samples in tap water at room temperature for 15 to 24 hours. Next is using any cattle tool. Carefully cut the seeds in one of 2 different ways. First is the cross-section or the transverse, and the second is lengthwise or the longitudinal. Then, the seeds after cutting. Second is using your pre-soaked seeds, sow at least 5 seeds. Then last step is examine the germinating seeds after 3 to 5 days from sowing. Then understanding how seeds germinate and grow into a healthy seedlings is critical for successful crop establishment. Seeds are immense biological importance. They contain high protein, starch, and all reserves that help in the early stages of growth and development in a plant. These reserves are what make many cereals and legumes major food sources for a large proportions of the world's inhabitants. So that's all. Thank you and God bless.